first rough cut series of the fall. Really glad to have you here. We meet every Wednesday at noon, and this year the event's at MSU only, but it's being broadcast as a webinar around to other institutions around the state. So happy to have our first speaker is Bruce Maxwell, who everybody knows. Almost needs no introduction, but I'll say a few words. He's a professor of applied plant ecology, which I guess is a mixture of agroecology and invasion biology. Bruce has been at the university since 1992. He came from the University of Minnesota, where he had his first job, but he actually got his bachelor's and his master's degree at Montana State University, so he has deep roots here. And all of us that know him know that he's a superb teacher and an excellent researcher and just a great colleague in general. And I've really enjoyed working with him on the wildfire pyre project, looking at pine invasions in the southern hemisphere. So happy to have Bruce talk. Thanks.
Thanks, Kathy. And I guess I should start out by saying that I'll emphasize the rough part of the rough cut here, in that the talk I put together is sort of a compilation of things that cover a big chunk of my career, and so I'm going to kind of pick pieces out. I'm also going to concentrate on the research that I've done in agriculture specifically, sort of to take this seminar series from more pure ecology, I guess, to a very applied aspect of what I do. So I won't be talking about the invasion biology stuff that I've done in the past. I guess I'm not in the way here. There's screens everywhere. So if I have to delay to point to something here with the pointer, it's so that people that are out there in cyberspace can know what I'm pointing at. I'm going to address three very disparate sort of themes in this talk, I guess. The value of models in conducting research, you'll just have to see if that theme actually emerges from what I have to say. I want to talk about weed impacts, a fundamental question in agriculture, which is it has sort of been the driving aspect of the applied part of my position. My position description says that I will, initially my position description was to look at the ecology of weeds in agricultural systems of Montana and come up with non-chemical approaches to management. And we'll see if I've accomplished that in any sort of way. And then I guess what it comes down to, a lot of what I'm doing more recently is really thinking about how to translate science or science-based knowledge into management decisions. And I think this could be indicative of any kind of natural resource management. I'm going to use the example of agriculture because it's one of the simple cases where the outcomes of management are very tangible, sort of economic outcomes. And so it makes it a nice system to look at how do we get science into management. Fundamental principle of weed science is that weeds compete with crops for resources and thereby reduce crop yields. This goes back to the Bible. People have known that weeds decrease yields. And so that's pretty straightforward. That means we need to remove the weed and our crops will grow. They compete in various ways for different kinds of resources. Plants compete above ground for light primarily. They compete for water and nutrients below ground. And so there's this overlap in time and space where they're sharing these resource pools, and therefore there's some sort of reduction in their potential to produce, and the focus species is crop, with the idea that learning something about the weeds and how they interact with these systems is a fundamental importance. So does that actually occur? And the theory that's been developed in weed science is that as you increase weed density, the crop yield decreases according to some sort of negative hyperbola, and that's been shown over and over with lots of different crops. And we have a nice nonlinear relationship that relates the yield maximum times some proportional yield loss here, and the I term in that parameter is the slope is this density near zero, and the A parameter is the maximum proportional yield loss. This F is simply the efficacy of some management that might come in. So if it's the idea that efficacy is 1, that means that there's no effect, and as you come down in proportion, so you can decrease that density effect and back up this curve is the idea for decreasing the effect of the weed on the crop. So we can actually extend that with a few constants. We take that same equation and then we feed that yield part. I guess I got to remember the point here. We take this yield piece and we stick this into this equation with some constants where we have P as the price received for crop, for a certain volume of crop. And then we have subtract off of that the net effects, the crop husbandry costs, and then we separate out weed control costs as H sub W. And with that, then, we can generate 2 different curves. The one is the green curve here is one with no weed control, and we get a net return that just mirrors the image that we get from the crop yield curve due to weed density. And we get a different curve when we do weed control. And with that, we can see the point at which those 2 curves cross, and that's the weed density threshold for triggering management if you're being an economically rational manager. So this is a nice way to say, okay, we can establish what the thresholds are for weeds in any particular weed species in any particular system, and then we can have a good recommendation as to when you need to do management, when you do not. So that's sort of the fundamental basis for a lot of the research that I was involved in early in my career. So the portion of this function that could be looked at in some consistent way across systems is simply this proportional yield loss piece, which turns into that particular curve. So now we have the mirror image because now we're doing yield loss. So now we have a value between 0 and 1, and we can say how consistent is that across systems. So one of the first projects that I was involved with really started when I was at the University of Minnesota was looking at corn yields, and this is velvet leaf density, one of the major weeds of corn in corn production. And what we wanted to find out after doing experiments from 1990 actually up to about 1996, consistently doing the same experiment where you would plant a crop at a certain density and then add weeds at different densities so that we get this stretch of weed densities. We could get the proportional yield loss curves and look at how stable those were over a number of states and years. And what we found was that, they're not very stable at all. Now our challenge became much more difficult. How are we going to account for this problem of instability? Certainly there's differences in years, which might make sense with different climates, different weather. But there were also this a definite site effect. So the weeds were not behaving the same at different sites. And what was that all due to? So that was a challenge to try to sort that out. Shortly thereafter, we looked at a similar kind of thing and say, what was the stability of the yield loss relationship across a number of states for winter wheat production, being the winter wheat production region, in response to jointed goat grass. That's what JGG is. And this work was done by a postdoc that was working with me, Marie Jesnick, who's now at Davis. And we found, again, real instability over years and times. And again, this was quite disconcerting. We thought maybe there was some relationship early on, but it just didn't pan out. So this was pretty discouraging that, gee, our whole job of finding these thresholds, these density thresholds, was going to be far more complicated than we initially thought. Coming back to Montana, did some similar kinds of experiments and found the same sort of thing, some real variability in the responses from site to site and from year to year. So this was with wild oats, which is our major weed in wheat production, dryland wheat production. So with that concern, how to attack that problem came down to kind of two possible research directions to follow. One was to really look at developing physiologically based models that would allow us to understand what causes those variations, first principle kinds of models. And that might be our way to really get at what's going on with these species, what's causing this site-to-site year-to-year variation. Another approach was to stay with an empirical based approach, but we would have to pretty much parameterize a model for every year kind of condition, every climatic condition, and we'd have to do it pretty much site-specifically because we had an inkling that history, the management history in any given field or any given place, played a big role in causing variability in this response of the impact of weeds. So we started looking at physiologically based in a very serious way, and shortly thereafter a paper came out where, with a really good data set in the UK, some folks found out that using the best models, or the models with the best reputation for doing physiologically based predictions, and we're really being widely used to say what might happen with climate change. This goes back, this is 1998 publication, this goes back a ways, that this was sort of the state of the art, but none of the models accurately predicted historical grain yields. And that was quite discouraging. So these models seemed to have all the right components. They performed fairly well for specific processes, but when it actually came down to predicting crop yield, the part that we really wanted to know about, they didn't do very well. So that was a bit discouraging. So we kind of backed off that idea and said, well, maybe we should go the other direction, look at empirically based, site-specific kinds of models and how we can maybe utilize those. About that time, we started getting information right out of fields, and we have combines with GPS on them, and every 3 seconds or so, they're giving us a yield throughout a field like this. So we're getting tons and tons of data. We have an amazing data stream coming in. And maybe we can relate that the yield information that we're getting there to not only the presence of weeds, but maybe all the other kinds of things that might be causing variation in yield. So we started following that path and thinking about, can we create experiments out there in those systems to get at this site-specific response and parameterize these fairly simple kind of regression models from these kinds of experiments. And with the technology coming along, we could actually put a map, a GIS map, into a sprayer here, for example, and do certain kinds of treatments out in a field, and then with the yield monitor get the response to those treatments. A lot of this could be completely automated. A farmer might not even know it's going on. So we tried that and worked out some ways to do that. There was lots, of course, details to making that successful, especially early on when the technology was fairly new. But these points, we were getting our yields, and we were getting enough yields in these different treatments where we would leave controls where we did nothing, and then would have other kinds of treatments, these other colors or other kinds of treatments in terms of weed management at that point, and looked at what kinds of responses we would get.
So are those herbicides, different herbicide treatments?
They could be different herbicide treatments. In this case, they were different rates of herbicides. So we could look at those responses. Boy, once again, we had an incredible scatter. I draw lines through there because we could get the significant linear responses, but oh my gosh, our certainty was still incredibly low. And even though we could overwhelm the system with data and the approach with data, we were still struggling with getting something that we would have some confidence about so that we could again come up with a prediction, a threshold, an economic injury level that would allow us to make a decent recommendation. Added to that, we started looking at, gee, where do we get different kinds of levels of yield? And here we have underneath here, this green area is where wild oats were present in the field. No density values here, just a presence absence. And then if you superimpose on that the different yields, the high yields being black or sorry, being red and down to black, and then the white areas were yields below 10 bushels per acre, which is basically non-economical production. But what we could see here is, this is a little disturbing because every time we get a high yield, it tends to be right where the wild oats are. That's and this is the place they were managed. Gee,
Everybody likes to grow there.
yeah, so everybody likes to grow there, and maybe the realities of what we've been doing in small plots in terms of planting weeds in with crops and getting a negative response, maybe that's all contrived. When we actually get out in the real world, it's not a real thing. It's not something, but wow, if that was the case, then people are killing weeds for no reason whatsoever, and they're spending thousands of dollars on doing that. So that is a bit of a concern. Again, data from that field, those scatters are kind of scary, and trying to come up with thresholds and sometimes getting double thresholds it seemed crazy. So weeds can compete with crops for resources and thereby reduce crop yields, or do they? Maybe we need to go back and address the very first principles of what we're discussing, or what we're thinking about here. So that brought on a study where we decided, let's take this apart. Let's go right down to the very fundamental question about how much dry biomass of individual plants can be accounted for by competition for resources. Develop an empirical data set, do a regression model, determine what are the factors that really contribute. And in parallel to that, use a simulation model that's based on competition for resources only, and use that as a process-based model to predict or simulate plant growth, and see if we can duplicate what we see in the empirical data set. So in parallel, we're going to use a simulation model. We're not going to parameterize the simulation model with our data. We're independently taking these two things to this approach to ask the question, does the theoretical process model produce data that will look like the data that we've produced in an experiment? So in a highly controlled condition in a growth chamber, we looked at spring wheat versus wild oat in monoculture and in biculture. We grew them in flats 50 centimeter by 50 centimeter by 30 centimeters deep, so they could grow for about a 12-week period. 120 randomly located seeds planted on a 1 centimeter grid. Each seed was weighed before planting, so basically we have a plant, we have a location for that plant, and we have a seed weight. We know everything about this system to start with. And we're going to let those grow. And we had a couple of replicates, and we grew them for 6 weeks and 12 weeks and harvested different flats at those different times. And then adjacent to this, we did individual plants growing in pots, and we grew those along just so we could see a free-growing plant, how does it grow? We could get a relative growth rate for those two species. The dependent variables were then the plant size, the dry biomass at 6 weeks or 12 weeks. The independent variables would be seed size, number of neighbors, total neighbor biomass, mean distance to neighbors, total difference in height, in height difference between the neighbors and the target, and then which is a surrogate for relative emergence time. And then we looked at those models for which variables would come in as predictors. So just again, to give you a sense from the diagrammatic sense, we're interested in a target plant in the center. We'd establish a certain neighborhood area or radius, and then we'd look at the number of neighbors around that plant as a predictor of its size, the biomass of these, the summed biomass of those neighbors, the mean distance to the different neighbors, and then the mean height difference of the neighbors, or the summed height difference. They were about the same. So in other words, we tried all the different formulations of how you construct a neighborhood to come up with what the best models were. And these were the best set of variables. And then we established what the best neighborhood radius was by where we minimized the Kaki's information criteria in those regression models. And where those were minimized is the point at which we were explaining the most variation. So we could lop off our neighborhood for each individual at that particular point. So for wheat, it was up here at 11 centimeters and 7 centimeters for wild oat. So what we did then is produce a data set from this box experiment. And it looked kind of like that. Way more variation than we expected. Once again, I'm always haunted by this. Sometimes I think it's just me. But I think it's probably the realities of biology is that we deal with scatters like this. So it wasn't a nice, it has a downward trend, we can see in that, but we thought it would be much more tight, given that we had controlled so much of the environment. But nonetheless, maybe the variation is due to different factors, yeah. Oh, I'm sorry, W is just the individual plant weight, and this is the number of neighbors in that 11 centimeter radius neighborhood. Thanks. So the full regression model that we looked at and to sort of weed out what the variables might be were these seed size, so the initial size of the plant, height difference, it gets the relative emergence time. If one emerges sooner, it would have an advantage in terms of getting the resources. Neighbor number, neighbor biomass, and the distance to neighbors. And what we found here, the colored in boxes means that they were significant parameters at both 6 weeks and 12 weeks within this monoculture analysis for the wheat and then the wild oat, doing each box independently. And effectively, we found all of those variables came in, except for the neighbor biomass didn't come into the relationship. Which for wheat, it did come in for wild oat. The other thing was that after 12 weeks, the wild oat seed really wasn't contributing much to explaining the size of the plant. It basically ran out of seed reserves, and therefore it was no longer having any impact on that on the size of the plant. That kind of made sense. The spring wheat seed being larger even after 12 weeks is still having some sort of impact on the size. The only other difference was the distance didn't seem to matter as much to the wild oats as it did the wheat. Most importantly, perhaps, is to look at how much of the variation we were really accounting for in the individual plant size, anywhere from 0.49 up to 0.63. And that was a bit discouraging to think that, gee, these are all the variables that we think should be explaining the size of these plants. So in this very detailed controlled experiment, we were really kind of puzzled by that fact. And then in the biculture, we did the same sort of thing. Now we have two different species, and we're trying to explain the target response to these two different species, the white ones and the green ones. From that, then we have a full regression model again, but it includes, oh, whoops, I went the wrong way. Now we have full regression models for both species in the biculture, and we have each one of them not only explanatory variables being not only those of the same species, but those of the other species as well. So the full models for each And when we look at what parameters are significant in this case, again, the colored in green blocks show the ones that are significant versus those that are not. And we see seed size is important throughout. The time sequence, the height difference wasn't so important to wheat, and we drop out the distance for wheat and the number of neighbors of wild oat. It didn't care about. It did care about the biomass of the wild oats, and so the distance of the wild oat didn't seem to be all that important. So basically what this is saying is wheat does pretty good on its own and doesn't really compete that much with wild oats, really. If you have a bunch of large biomass or large plants of wild oat in the system, it can certainly have an impact. But that's about it. Wild oat, on the other hand, is quite sensitive to the spring wheat and as well as its own density. And so again, that it sort of starts to tell us the story of, okay, what are the things that really count in this neighborhood of interacting plants? Again, I go over here and say the R squared, however, is relatively low. We're just not accounting for much of the variation. Where is that other 50% or 40% of the variation? What could be causing that in this system? So the disturbing conclusion is seed size, relative emergence time, and neighborhood conditions only accounted for 50 to 60% of the variation in individual target plant size. Yeah.
I mean, at that time, and maybe you'll go through that later, but as a microbiologist, actually, I would say it's a very nice value. I'm surprised it's that high.
Well, except under the, I guess we were after the holy grail. We should be able to explain all of the variation.
We're not accounting for what I would think is the most likely cause of divergence, which is that all those plants are not genetically the same. So,
oh, you're stealing my thunder.
That's what I said maybe after, so I didn't want to go there yet.
No, that's good. It's a good setup. Damn. So let's turn to, in trying to say, okay, let's see what happens. We got this simulation model that's based on competition for resources. Let's look at using it to produce our data set. I'm not going to go through the details of this, but basically it draws on Dave Tillman's initial models. And I'd worked with him in Minnesota on some of these early on models that were coming in. Looks like we're getting some messages. And the idea is that this is sort of the fundamental resource competition kind of model, and we kind of group all the resources into one category of resources. But generally, we can say the kinds of things that drive this are the amount of resources available for growth, and that we get a particular kind of growth rate from that. And the way that we look at how resources are used are based on the size of individual and how far away it is from its neighbors. So very similar kind of approach. The simulation model starts with a seed size and then simulates the growth of each individual plant in terms of dry weight over a number of weeks. And we found out that with this model, we could do the exact same analysis. This is the kind of data, and in other words, this is individual plant size, this is number of neighbors in the neighborhood, and we get a data set that looks roughly something like the one that we saw empirically derived from the box experiment. And sure enough, the neighborhood radius that we set in the model at 10, that's where we got the best response in terms of figuring out how many neighbors to include in a neighborhood with a particular model. And we found that the best models were somewhat similar to those that we were getting with the empirical data set. But the interesting part here was that even though competition is implicit in the model, I mean, it simulates growth based on competition, we still only describe 75% of the variation. So we thought, oh, well, the target wasn't maybe 100%. It really should have been 75% because when you put individuals of different sizes to start with together in a system, there's going to be variation in this response, even though we assume everything else is consistent. But that means that our empirical data was still missing then. Instead of missing 40%, we were really only missing about 10 to 20% of the variation. So where did that come from? And, well, even when competition was fixed, this is what I just said, 75% of the variation in target plant size was accounted for. Only 50-60% was accounted for with the seed size, relative emergence time, and neighborhood factors with the experimental data. 15-25% of the variation wasn't there. But when we went to our growth analysis experiment, sure enough, we found that about 11% was due to variation in relative growth rate just between individuals. So probably genetics. Clearly that's the way it looks when you go, the crop only had 11%, but the variation from the wild oat was like 20%, twice that. So the weed was more variable. So we're coming pretty darn close now. We're feeling pretty good about the fact that, okay, maybe we can conclude this. Weeds do compete for resources and will reduce crop yields. There really is something to this. Now, how do we scale up? How do we get this kind of information into our model so we can be more predictive when we go back up to the field scale? So, how do we parameterize these models is when we're dealing with all the things that we know we're trying to understand in these systems. Well, the land-grant system developed this wonderful approach to understanding our system and did quite well for plant breeding in that we could plant all these different new tested varieties around the state, and sure enough, we could predict pretty well what the best variety was to grow here or here or here in different parts of the state. But when it came to more of these biological kinds of ecological interactions, this didn't work well. So small plot experiments were not going to be the way to get to this parameterization of what the impact of weeds on crops was. That became very clear. So we implemented the concept of adaptive management. Let's go and let's see if we can set up these experiments on fields and try to understand what the variation is in responses in those systems. So our approach has been, okay, there's a whole lot of things causing variability in all the different things that we do out there. So can we take information that's coming off of our machines and off of satellites, et cetera, get yield information, weed presence or weed density information, seeding rate information, fertilizer information, somehow synthesize that in the black box, the proverbial black box, and then give some sort of input prescription or some sort of recommendation as to how farmers might behave in an economically optimum way. And at the same time, perhaps this could actually deliver the ideal experiment to do out there in order to parameterize these models. And then this becomes a nice circular sort of approach where you might use a Bayesian statistical updating to actually get this to generate different kinds of responses. And get a sense for, over time, in any given field or even portion of a field, can we start to gather enough information to empirically derive what the best predictor of a response in that field will be. So then we tackled all the major problems that we think are causing variation. So we've been working in a number of fields, but most intensively on one farmer's field, Chuck Merja. This is Power and Vaughn. This is just north of Great Falls. And he has a few fields where he has been collecting data since about 1994 on yield information. He's had yield monitors. He also has a protein sensor on his machine. It's all this different kinds of data that we're getting every few seconds. And so that's all really useful. And he's also mounted a camera on the front of his sprayer so that when he goes through the field, when the plants are in the seedling stage, he's basically getting an NDVI measure every few seconds. So there's a bunch of information that we can give.
NDVI? What is NDVI?
Oh, it's, what is it? Normalized difference vegetation index. Is that right?
And
In English? That is basically a reflectance. So it tells the greenness of the field. Is that fair? There's people who know much more about it than I do. But, yeah. So we set out to an example is developing some of the equations and what are the best predictors of these that come into particular fields. And basically we have this long list of variables and we sort of weed out what those variables, the best variables are. And we see things like elevation telling us something, so that gives us some idea of the configuration of the field. This was soil water. In this particular field, we had actually had some soil moisture measurements. That's not a very common factor, but sometimes we are able to get that. We're looking at different ways of getting that from the precipitation history currently. But anyway, that's included. NDVI, the Barris, which is electrical conductivity. It's a machine that gets pulled through the field and basically gets this continuous measure of electrical conductivity. Yield from a previous year, soil nitrogen at a 24-inch depth. Again, in this particular field, we had an intense set of soil samples that went down to 24-inch depth. Now, this all seems like this is not very easy to gather data, but in fact, there's being, as we speak, they're developing a machine that'll probably fit on a combine that'll take a soil sample every few seconds, and won't be long, and we'll probably have a lot of information about the soil sample information that we're getting as well. So these are all things that are all
How did they get the soil nitrogen at 24-inch depth without the new machine?
How do they do that?
I mean, you just have to take a sample at 24
They
inches.
take a sample, they just drill a hole as they're moving along. So that's, it's in development, but it's looking promising. Let's see. Oh, so then we can predict a yield from those variables, and it's against the observed yield is a reasonable approximation, let's say. So you can kind of see spatially how that works out. The interesting part was, especially when this was funded by the Fertilizer Tax Fund, was that urea wasn't an explanatory variable. So in other words, the urea fertilizer, the nitrogen fertilizer that they were applying, didn't help predict yield at all. And so that was a difficult one to explain to that group, but we somehow continued to get funding from them to do some of this preliminary work at least. The farmer, interestingly, was onto the idea that you could put variable rates of nitrogen on out there, and he should be able to detect those with his yield monitor. So that's what these strips were some high rates of nitrogen that he put through the field here, the darker areas being the high rates, but they really don't show up in that observed yield. So that was kind of interesting in itself, and so again, even the response to nitrogen seems like a very straightforward one is not so straightforward. Given that, there's all these other kinds of variability that we have to deal with as well. Variability in input costs. This is the price of urea in dollars per ton. This is the variability in price received for wheat. And I'm going to look at just, if we assume that this is the price range that we might get for wheat, what we find, the optimum, the amount of nitrogen that you might apply on the same field, given different prices received, changed tremendously. So the red areas are the maximum amount of nitrogen you would want to put on, and the blue areas is no nitrogen putting on. So if you're going to only receive $8.50 for a bushel for your wheat, it says you shouldn't put on very much nitrogen at all, only in one little portion of the field. If it jumped up to $9.50, it said max it out. Put on all the nitrogen you can, because then you'll get the maximized net return.
Didn't you say that urea had no impact?
It did on that particular field. Now here, in this case, you can, if you go place to place in the field, you actually can get a response out of that nitrogen using the model. It will predict what is ideal, even though it doesn't come into the model on the other field. Any given year it might. So I think I've got the, oh, I didn't include that. This is on a different field, but nonetheless it has what you see, as much variability as we see with the price, you're going to see from field to field and from year to year. Yeah.
So,
We have several people on remotely, and they can't hear your questions, so if you could push the front.
I don't have any written in front of me.
I'll try to repeat it.
So have you guys messed around with anything like anhydrous besides urea?
No, we just looked at urea just to, yeah, because we just haven't had the resources to go beyond that. So it's sort of a first approximation. So the question was, have we looked at any other kinds of fertilizers? There's anhydrous ammonia as well, used as a common herbicide or a common fertilizer, but we've just looked at urea. So this is basically a different field. I am jumping around here a bit on that, but the point was that the kind of variabilities that can come from some of these other factors are the ones that tend to be driving more of the decision process, or should be driving the decision process, really, than maybe the biological interactions even. So we learned the variables that will most accurately and consistently predict yield and protein content under constraints that they are easy and cheap to obtain, were NDVI, elevation. We did have nitrogen rate. Now we had to kind of cheat on this in some cases. If we did this as an average overall field, we'd typically find a nitrogen rate probably wasn't consistently coming in. But again, we were being funded by folks that wanted to know about that, so it went into the model regardless. Previous year's yield, the presence of weed, and precipitation has become our target. We haven't developed a great way to bring that in, but we're convinced it needs to be in the model, and certainly gives us the year-to-year variability that we're looking for. So we produce yield, and we produce a model for protein as well, because that's the things we get paid for. The nitrogen fertilizer, as I said, rates are extremely sensitive to price received for the grain, and that makes a big difference in the economic response in a system. So the next level at which we've sort of tackled this problem is, again, using a simulation model to ask the question of what's the best field scale site-specific parameterization. Is it possible to parameterize a model for a given field? So this is a field here, and this shows year one. We show the yield response that we predicted with our model, a set of treatments that we would overlay on that field, an observed yield from that field, and then a net return, a profit that would be gained given what we did on that field or what it called for. And then progressively, as we get information from different years, how long does it take before we actually stabilize those parameters so that we get good, solid predictions that we feel like we can make good recommendations from? In other words, we start to reduce the variation in what we see in the system. And we found that after about 6 years, this work that Patrick Lawrence has been doing, it takes about 6 years in the simulation for us to stabilize, given the kind of variation that we tend to see out in the real world data anyway. So again, this is a parallel use of a model, a simulation model, to ask sort of the general question about what's the best approach to perhaps take in terms of developing the science behind good recommendations. And we can take that a step further and start to say, once we have a model like that for a given field, now we can start to say, what's the resilience of a system? So if we go back in time and use the weather data from a drought period here, we can say, what sort of management strategy, these are different applications of nitrogen if we did them on a full field basis, which ones would pay off or which ones wouldn't, or which ones would, more importantly, make you go broke. And we found that if you went to a site-specific kind of application based on our optimization routines, that in fact you were going to be safer, you're closer to the zero line than any of the other particular management options. So, and it turns out that during this drought period, if you look at the number of bankruptcies per 10,000 farms, they certainly were peaking out at this time when we were moving into that drought period, and farmers were suffering tremendously, primarily because they were following these management approaches, which were sort of consistent full field management approaches that were not getting them any response in their system. So that's the kind of thing that we do. So in summary, what I like to present here is the framework for modern decision-making in agriculture and how we try to integrate different kinds of information into a decision and how important that is. And the process has really been fairly interesting, and it really includes a very important social science aspect to this, because how do people learn, and how will they utilize data in the end? We can produce the best models in the world, but if people aren't going to use them, you know, it's kind of like producing a toaster for people that don't have electricity. You've got to kind of walk them into this place where they can get their head around how to manage, and respect the traditional knowledge, the casual observations that most of their management decisions have been based on for a long time, and somehow integrate those into a process that's more science-based in this shaded side over here. So with the fact that what we do is produce information that is not totally prescriptive or deterministically prescriptive, but more importantly is probability-based, and that those probabilities, we have to teach our farmers, our land managers, that you get a certain probability. We know you're gamblers out there, so what's the best way we can make you the best gambler in making the appropriate decision? And how do we pull that into a framework of understanding that will be helpful? So hopefully I've demonstrated that how through a long period of thinking about systems and how we make decisions and the kinds of fundamental processes that are involved, how we think about that and how that might relate to natural resource management and inclusion of an adaptive management process within a framework or something like this. Okay, I'll stop there. I'd be happy to take any questions. Yeah.
If you guys could go through and just turn these lights on, then we won't touch the front of it and the green light comes on. So anytime you see a mic, if you could turn it on.
So I know this can get you quickly mired in minutia, but I'm wondering if you've looked at the structural integrity of your model. It didn't look like your model included any interaction terms, and I was wondering if there's a reason to think that all of those independent terms are going to have a linear effect.
Yeah, no, there's lots of nonlinearity in there. We did look at interactions as part of the straightforward regression. Mostly they were rejected in those models. There's a lot of autocorrelation in there, but we're still in a process sort of winnowing out what the best full set of parameters might be, and realizing that we should carry that into any particular field, and let the model be developed simultaneous with just as the data comes in. Okay. So in other words, keep looking for the best model through that process of a 6 or 7 year period of getting different information that's site specific. It's quite possible that, and we will include in that interaction terms. Okay. We've avoided some of those because of the complexity of how then you deal with those in the end product, as you are probably well aware of. But it's granted, it's an aspect that I glossed over here, but we have done a whole lot of thinking about how do we get that best model.
Okay. Wondering about interaction with other, like, nutrients. You got all these soil samples. Is there any interest in anything else? Zinc, iron, I don't know what it might be.
You could start to add all those kinds of things into
Do you have the soil samples still, or are they all gone?
Oh, no, those soil samples are gone. Unfortunately, of the ones that we sort of artificially had, and we only had them in one field 1 year. So that was the particular year that we thought, okay, that's the most full data set and different parameters that we could put in the model. Let's see how that works and which ones come in. Looking to the future, that at some point we probably will have a nice stream of that kind of information as well. It could definitely be helpful. It should also be helpful as we start to market our crops differently. I think as we start to market our crops here, we market our crops with protein as well as just yield. We can imagine that we might start to monitor for all kinds of other qualities of those crops as well that might start to get a premium in some different markets. So I think all those kinds of things are going to be really important as we move into this age of a useful data stream coming out of that.
The sensor that gets protein, is that near infrared? You know?
Yeah, I think it is.
There might be other data, information in there. There might be other information in that data.
Oh, probably is. Probably is. And those monitors have been they've been worked on for about 8 years, I would say, where they actually have had them out on farms.
I hadn't heard that.
And the information is stabilizing. They had to deal with a whole lot of things. You know, you can imagine all the dust and all the stuff. The yield monitor gives you a moisture measure of the grain as well. Yeah, and actually that's one of the variables that we're using. Yeah, Kathy?
Could you help me connect the dots in your talk a little bit? Because you had a very plot-based experimental study, and then you had a single field where the sort of recommendation was that fertilizing doesn't make that much difference. And then you went to pricing at a really large scale. And so what did you learn from looking across those three scales
well, it's a place where we can, I'll start with saying that it's a place that we can tackle some of these scale issues, I think, and the disparity of kinds of variables that cause variation or uncertainty in any particular response. And so I guess what I was trying to say is we've got variability at all these different scales, and we've tried to quantify that variability in a way that the patterns that we see in that variability we can use to describe the uncertainty around any particular recommendation. And some recommendations or some prescriptions or some management approaches are naturally going to be much more uncertain than others. What's interesting is what we've thought all along is that inputs basically decrease uncertainty, increase certainty, because they suppress variation in our system. That's not consistently what we're seeing. And in fact, they might actually elevate it in some cases. So that's an interesting generalized finding. I don't know, I think given what we've done so far, it would be hard to publish that because that is so contrary to agricultural research in general that it would be difficult. But I think there's some good reasons for that, and sort of this interplay of the simulation models to work at what is driving these factors in parallel with some of the observed data that helps look at where do we get data that looks similar, sort of the likelihood approach of understanding what the patterns are in that data and the uncertainty in that data. That's how we'll learn more about that process and about what makes those connect at different scales. Certainly you have, pricing is established by markets, and right now it's a random sort of variable. All we can do is use chunks of history and say that this seems to drive the price of crops. It turns out that might be more predictable, and working with economists, that might be more predictable than we think. There are certain things that drive prices up or down based on how great the wheat yield was or protein values were in Argentina or the Middle East or Russia. So there's these kinds of things that play a role. So there might be some level of prediction in that. Again, really a global scale. Now you come down to this, we're going to jump right down to a field scale. So, okay, what makes the difference here? What drives nitrogen costs? Well, it's natural gas, and it's basically the willingness to pay of corn and soybean farmers. Oddly enough, because that's where the market is. So it's kind of interesting. Wheat production is sort of a second cousin to all the other production systems in the country because there's so much more money to be made in those systems from an input point of view. So there's things like that, these side effects that are out of the control of the farmer. So when they make a decision, that's why you have such huge error bars on what the general, what you're confident about where the relationship is going to exist, or where that, general trend might be. Does that help?
Yeah, thanks.
Okay.
I don't know how much time we have left, but I just had one question, and maybe most of you, because it looks like you all are plant scientists, or almost.
Yeah.
And what's the incentive for farmers to have all these monitoring devices? I get now that they want to know how much protein is there as they collect the crop, because that's what they use to actually sell it. But why would people start hooking cameras or more sophisticated devices on their machines to collect data? Of course, I can see why they would want, in theory, do that, but it's certainly a costly apparatus. And as we see now, we are still working out how to use this data to help them. So why would they do that? Are they just in collaboration with MSU or with other universities? Do they receive money for that? I'm just curious, because I'm surprised by how much data you got.
Well, we work consistently with about 5 different farmers that are sophisticated enough to have a lot of these monitors and things, and are gathering data. It's a hobby to them. They're not making. They went for a long time without making any money off of it, and some they still are struggling to some degree.
Do you think
as to whether it's of value. They see that they have the same vision that we do, that this is going to pay off, and having data, a backlog of data, is going to pay off. Mostly though, they're just techno geeks. They just like stuff. They like to farm from their computer. They're those kind of people. I mean, Chuck Merja that we work with is a Stanford-trained engineer and was working in Silicon Valley when it dawned on him that he'd rather be a farmer in Sun River. So,
if I was to become a farmer, I would probably do that.
Yeah, right. Many of us probably would. But it takes a certain kind of person at this stage. These early adopters are people that are basically funding this on their own back. Although a lot of the companies developing the monitoring or monitors or the information devices, I'll call them, have clued into these people, and they work with them very closely.
Is there grants, for example, from the USDA to do that? Where I would say, I'm a farmer, I'd be interested in monitoring my field for research.
The only way that that kind of thing's being funded is through SBIR grants. Do you know what those are?
Yeah.
Yeah. So that's the way that it's being developed. Yeah.
One more quick question.
Well, maybe we should break it.
Okay. I'll be happy to talk to you.
Oh, thank you.
Thanks, everybody.